So it looks like now you can start.
Okay, shall I start? Do you see my screen?
We can see your screen and we can see your face in small. Okay. And we can hear out without echo. Let's see.
Perfect. Do you also see my mouse that I move here?
Yeah.
Yeah, you see my mouse as a mouse pointer? Okay, good. Then maybe we just get started. We are like 10 minutes behind time. So maybe I rush straight into it. Okay?
Okay.
Good. So then, thanks for joining my talk. So I have like it's two parts, right? So like I prepared it as a two-part presentation. So it's like the whole session is about privacy preservation and about systems medicine, and I have two parts. And in the first part, I want to talk about what I have learned about Arnold Schwarzenegger while I was younger and studied the survival of breast cancer and breast cancer molecular diagnostics. So we will get to there in a little bit. So first, a little introduction about myself. I'm Jan Baumbach. I am a professor at the University of Hamburg, and I just moved from the Technical University of Munich in southern Germany, in Bavaria, to Hamburg, so from very south to very north. Where I'm now a chair of the Computational Systems Biology department at the university. We host 3 laboratories, so like our acronym is COSI Bio, like Computational Systems Biology. We host 3 labs. We are about 30 researchers. We have a couple of administrative and IT staff. And you can find us online at cosi.bio, right? If you go to cosi.bio, you will see, you will find our institute online. As I said, I'm just one of 3 labs. So I'm the head of the department or the head of the institute or the director, or whatever you want to call this. But we have 2 more groups. One headed by Olga Zoy. She's mainly analyzing genomics and transcriptomics data at the CGET group, and we have Olga Zolotareva as group leader, and she is focusing more on mass spec-based systems medicine, a lot of proteomics, metabolomics, but also working a bit on this kind of transcriptomics data analysis. Good. Let's get straight down to the topics, right? So what motivates me, and this is one of the plots that I found extremely striking when I saw it the first time, maybe like 7 years ago was the first time I saw this. So this is the top 10 drugs prescribed in the United States today, right? And those guys here, right? And the corresponding indication. And then there is something that is called the numbers needed to treat, short NNT. And the numbers needed to treat essentially is that number of patients you have to treat such that one patient shows a significant benefit. So in a perfect world, you would like to have this NNT to be 1, right? So you go see the doctor, you say like, oh, I have arthritis, or I got high cholesterol, or Crohn's disease, or multiple sclerosis, or whatever, right? The doctor prescribes a medicine, and you would expect the medicine to give you a significant clinical benefit, right? So that would be the case if you would have an NNT of 1. In practice, it looks more like this. So the blue guy is the one that benefits from that corresponding drug. And the red guys is the people you need to treat until you see one such blue guy, right? So this is the numbers needed to treat. And this picture is horrible, right? So you have tons of people, independent of which of the top 10 prescribed drugs you take. You have a hell lot of people that take the drug, but have no clinically significant benefit from taking the drug, but in the best case, not suffers any side effects, right? And the question is, why this is the case? And part of the reason is the way we do medicine nowadays, right? So we still work on disease definitions that essentially go back to the 19th and 20th century, right? But many diseases are defined by the name of the doctor, right? Like Huntington's, right? Or Parkinson, right? Most diseases are still organ or, worst of all, symptom-based, right? So take cardiovascular diseases, take hypertension, right? Or chronic kidney disease, non-alcoholic fatty liver disease, and so on. So they are organ-based, right? So if you take whatever hypertension, what does it mean? What kind of diagnosis is that? Well, it means you have elevated blood pressure, and then of course you can give a drug that decreases the blood pressure, and then of course the symptom will disappear, but you're just treating a patient, but you're not curing the patient, right? So, and this is of course a problem in the long run. And well, like one way of narrowing down on this problem is the so-called network medicine or systems medicine way, where well, you make such graphs here. Each node in a graph corresponds to a disease, and an edge between two such nodes means these two diseases have something in common. So this picture that you see here, like this graph in the background here, is coming from Laszlo Barabagy, the famous dizzyzome, right? So I call it the gene-based dizzyzome, right? So essentially what he did is he checked GWAS compendia, and whenever two diseases have at least one disease gene in common, given like genome-wide association studies, right? One associated disease gene in common, then he's making such an edge, otherwise not. Then of course you can make these edges weighted, right? And then you can cluster such networks here. Then you see that they fall into cliques, right? And then you can have a look at these cliques, right? There's different ways of making such networks. Another way would be to look at comorbidities. So I preferably look at comorbidities because they are more bias-free, right? But anyway, so then in a comorbidity network, you would check if you would make such an edge when the likelihood of seeing a disease 2 given a disease 1 and the other way around is above a certain threshold. And if you see something like that, then you make such an edge, otherwise you don't, right? Okay, but either way, independent of how you look at it, whether it's disease gene-based or whether it's comorbidity-based or whether it's based on joint symptoms or any other way you could think of, at the end of the day, such disease clusters emerge. And if you look at these disease clusters, you will sometimes see things that you would intuitively find very surprising. If you take the gene-based dizzyzome here and look at such clusters, you see several of those, and sometimes you see stuff like stroke, Alzheimer, migraine, dementia, right, being in one cluster, right? And then the question is like, why would this be the case? So like, right, like what is there any molecular understanding and any molecular cause for this, right? And that is called essentially like network medicine. If you look at it top down, right? So you start with the diseases like this here, the disease networks, right? And narrow down on disease clusters and try to look for something that is similar between diseases, right? So it's called joint disease mechanisms, right? And there's a growing body of literature trying to go down this road and try to identify such disease pathways, right? That has been very successful in oncology, right? So it's widely accepted that cancer is a disease of pathways and that you want to treat it this way, right? However, in most other fields, this is not so widely accepted, or we are just in the beginning of understanding what's actually going on here. So if you look at it top down, so you start from such disease clusters, then it's more called network medicine. If you look at it bottom up, then it's called like starting with omics data, gene expression, protein expression data, right? And look for something, some mechanisms, right? Which essentially here like some pathway where an aberration or a disequilibrium in whatever molecular pathway gives rise to the disease, right? Then this is more called systems medicine, right? So systems medicine is more like bottom up, network medicine is more like top down, if you want it like this. At least that's my definition of the two fields and how to differentiate them. either way, at the end of the day, where we want to go is we want to move away from defining diseases only by symptoms or organ or whatever, right? Like the name of a doctor, right? We essentially want to define diseases based on the molecular causes. So there's any kind of either individual ones or a mixture of symptoms that emerge because of disequilibria in molecular pathways, right? And that is essentially what we actually would like to use as disease definitions. But that is tricky. Okay, so I dedicated a lot of research, many hours and like weeks of my life to this topic, like to this field of systems medicine, identifying such mechanisms, developing computational tools to extract or to identify such mechanisms from omics data, to find such disease clusters, find commonalities and differences and so on, right? And what I do is, like, every year I go with the whole lab, we go on a retreat, right? So like we travel somewhere, right? And everyone PhD student and above, PhD students, postdoc group leaders and myself, right? So we all give short presentations, like 15, 20-minute presentations. However, we do not talk about our own work, right? Because we know what we are working on each other. We have regular seminars and so on. We have weekly seminars and whatnot, right? So like the people know what I'm working on. I know what the people in my lab work on and so on. So no, I asked the people in my lab to give a research presentation about a research topic of their own choice, but their own research, right? So they can talk about whatever they want. It has to be a scientific talk, but like, so, and it wouldn't harm if it's a little bit funny. And you have no imagination how cool talks people have there, right? About the effect of prolonged space travel to the human body and whatnot, right? So we had a project where we had developed one of the first AIs that could play StarCraft against each other, and an autoencoder that made Where's Wally pictures from a given set of Where's Wally pictures and so on. So one of these at one of these retreats, like the last one before Corona, we went to Reykjavik on Iceland, and we had our retreat there. And Simon, he is now a postdoc in Copenhagen, but back then he was a PhD student in my lab, gave a talk about breast cancer and gene panels that predict breast cancer outcome. Right. So when I ask what I learned about Arnold Schwarzenegger by studying breast cancer, I should actually ask what I learned from Simon about this. And the next few slides that I'm going to show you are indeed slides that I borrowed from Simon, right? So that is actually the next couple of slides essentially emerged from Simon's talk and retreat talk back then. And I'm showing and presenting them to you now because they illustrate a big problem that we have in systems medicine and in the whole systems biology field. Okay, so what is the idea? The idea is we want to stratify patients into groups of outcome, right? So good and bad outcome. Yeah, let's say tumor, like survival, 5-year survival, yes, no, is one way of doing this. Or a tumor recurrence, yes, no, is another way of doing this, right? So like we have a good outcome, we have a bad outcome, a typical case control study. Okay. So, and we want to identify a set of genes or any kind of marker panel, right? Also called a signature or a profile, right? So however you want to call it, it's usually genes based on gene expression or mutations, right? So you want to identify a set of such genes, let's say, right? Here, that's what we have here on the left side, right? And based on this set of genes, you can distinguish good from bad outcomes. So what you usually do is, I mean, there's different ways of doing this. Use any kind of clustering tool, right? So we take such the expression, for example, of a small set of genes. Then one way could be you do a principal component analysis. You split your data on the first principal component, right? Then you have your two groups, right? And then based on these two groups, you can run your Kaplan-Meier statistics. Here you can make these Kaplan-Meier plots here, right? And you can even give it a p-value, let's say using the log rank test. That would be the typical way of giving it a p-value. And when this p-value is smaller than 0.05, right, then the people are very happy because they say, hey, I have identified a marker panel, in this case, a set of genes, right? And based on this set of genes, I can identify good from bad outcome significantly. So, and then people publish this and say, hey, cool, there is, I have a gene marker panel that I can use for diagnostics. Okay. Well, there's many such gene panels out there, really, really many, right? So here I just show you some for breast cancer. So this was one. Yes, more papers about breast cancer gene panels. So in all of these publications, and yeah, Simon really did screenshots of all those. In all these publications, some authors claim, this is now Nature, Cancer Cell, Nature Genetics, right? So in all these papers, the authors claim to have identified a set of genes that predict a breast cancer outcome significantly, right? Whee! Cool. So Simon said, okay, well, why not adding 3 additional such gene panel signatures to the gene pool, right? So I show you just one of the 3, right? So now my slides are a little bit different, right? So here showed us 3 such slides or 3 such gene panels. I just show you one, right? And my favorite one, and it's called Arnold 255, right? So if you're interested in the other 2, they're called HP40 and Pikachu star 5. I can show you either in Kyiv or you can ask me later. I have them as supplementary slides. Okay, so Arnold 255 was Simon's first gene signature, first gene panel. It consists of 11 genes, right? It's those genes here, right? And when you take the gene expression data for these genes here, right, run a principal component analysis, split on the first 2 principal components, then do your Kaplan-Meier plot here, right? Then do a log rank test. Then you see that the log rank test is significant. P-value is smaller than 0.05. You see a difference between the 2 groups he's looking at. I'll give you more details in a sec. So we were like asking, hey, Simon, give me a break. So what is it about? And he was like, yeah, this data is coming from a time series mortality statistics from an Austrian cohort. We were like, okay, what does it mean? Right? So essentially, that is what he did, right? So he went to TCGA, the Cancer Genome Atlas, and to the breast cancer project. He obtained RNA-seq data from about 500 patients. And he also downloaded the survival times, okay? Then he split the data into good and bad survival, right? And now he needed a gene panel that distinguishes between this, right? So how would he get to such a gene panel? So what he did is he went to moviebodycounts.com and entered Arnold Schwarzenegger. That gives you a list of Arnold Schwarzenegger movies and the body count, which is the number of kills in a movie. So what he then added, he added 255 to the number of kills in this Arnold Schwarzenegger movies. That is those numbers here, right? And that is the genes he extracted. So he worked from that moment on with gene numbers 343, 332, 326, and so on. So why did he add 255? Well, because he wanted to call his gene panel Arnold 254. Right, and the reason for that was he said is that the real commercially used gene panels, so people became rich with such, right, have these fancy names. They're called like PEM50, right, or Mama Print, right? So and he wanted to have something that sounds equally cool. So he said, okay, I call my gene panel Arnold 255. So he had to add this 255 somewhere, right? If you're a computer scientist, you might understand also by 255, right? It's a magic number for computer scientists, right? So he added 255, and that is the genes he worked with, right? So, bam. And remember, right, when we use those genes, gene number 343 in the data set, 332, and so on, we can make a Kaplan-Meier plot, right, that for the 2 groups of patients, those with the good and those with bad survival, right? And we can make a log rank test, and the p-value for this is significant. In other words, what he did is he found a fancy or funny way of making random numbers, right? Of selecting random genes, right? And with these random genes, these 11 random genes in this case, right? He could predict good from bad outcome in breast cancer significantly, right? We were about like, whoa, right? So most people in the lab were shocked and said, like, well, so there must be a mistake, right? So I could already see where he's having that from, right? So every PhD student starting in my lab has to read this paper here, right? And back then, I also gave this paper to Simon. It's from David Bennett and his colleagues, right? And essentially, yeah, you can read it yourself. So it says most random gene expression signatures are significantly associated with breast cancer outcome, right? So what they did there is actually quite easy. So they take the MSigDB database for molecular signatures, right? They generated random signatures, that is such gene panels of the same size as the original ones, right? Then they performed the analysis that I described. So they just do a PCA, split on the first principal component, do a log rank test, and check if it's significant, right? So, and then they see that in MSigDB, about 67% of the signatures have a p-value smaller than 0.05, and 23% of smaller than 10 to the minus 5. Okay. However, you need to compare this to random gene panels of the same size, right? And here you see that you have a higher chance by picking random genes, right? Of a same number of random genes, you actually have a higher chance of achieving these p-values, or let's say roughly the same chance, a little bit higher. And of course, this is a problem, right? So if we come to breast cancer, just to finish up the story about breast cancer here, right? So we had a closer look. Then only 38% out of the 47 signatures to predict breast cancer progression had a stronger association to the outcome than the top 5% of the random gene panels of the same size, right? And well, what can we conclude from such a jokey, from such a funny like presentation here, right? So you not just have any professor from Hamburg complaining about gene sets, no. So we can actually learn something from this, and what you can learn is that the explanatory power of gene sets per se is comparably limited, and there's several reasons for this, and one reason is that we only work on the seized genes here and not on the seized mechanisms, right? So you can imagine, let's say you have a couple of driver genes that are, but for cancer, for example, that are mutated, right? So, and these driver genes, they don't act alone, right? So they're not acting alone. So they're acting in an orchestra, right? So they're connected by other genes. And then, of course, the problem is that these genes react to the driver gene, and then they are called passengers, right? And now you have passengers that behave differently, and you have other genes in the neighborhood, in the pathway sense that react to these passengers. So you have passengers of passengers, and it takes a while until the signal dilutes over the network, right? So long story short, this mechanism, this stuff here, right? This stuff here works because gene sets, right? The sets themselves, right? May purely consist of passengers, right? In other words, what you do here is if you just pick genes that are significant for something, you're comparably likely to pick any gene that reacts to genes that react to genes that react to a driver. So in other words, you're likely to classify with a surrogate marker, which is not a big deal if you just want to predict something like progression, right? So in this particular case, all these gene panels, they still make sense. I mean, you probably only need one because they all correlate with each other, right? So because they in any way correlate with the driver. But if you want to understand anything about the disease, and if you want to actually like get down to curing it and developing drugs that don't treat the symptom, but like address the cause, then you need to get away from such disease genes and need to go down to mechanisms. So here's another example on Huntington's disease So here you see gene expression data set, right? So we have the like genes taken from the or samples taken from the caudate region of the brain in Huntington's disease patients and in a case control fashion. So here we have the Huntington's disease patients, here we have the control group. And here's just the first, what, I don't know, 50 genes, right? So in reality, this is 22,000 genes, right? So if you have 22,000 genes, but only, what, 100 patients in each group, right? But 22,000 features, right? If you do a standard statistic, you will always find at least a couple of hundred, if not more, genes that are significant, even if you correct for multiple testing, right? So partially because of statistical problems, right? I mean, you just overfit, right, in a way, right? However, the other reason is that you have passengers and drivers, right? And you have way more passengers than you have drivers, right? And you cannot distinguish per se passengers from drivers. So one way to address this is to map this expression data to networks, right? So here is just an example, like the protein interaction network. This is the human interactome network from 2011. Now it's way more dense and way more rich, right? So you can get it from BioGrid or IID, right? So every node here is a protein, and an edge means you have a physical interaction between two proteins, right? So what you're now looking for is any kind of subnetwork where genes are, or that is enriched with differentially expressed genes in most of the Huntington's disease patients compared to the control group here, right? So there's several tools for this have been developed over the last years. So I have been in this business for quite a while, and one of the first tools we developed that actually worked is called Key Pathway Miner. It's now one of the standard tools. You can just Google for it. So what Key Pathway Miner gives you, it extracts a maximal connected subnetwork where all genes, but a certain number of exceptions, let's say k equals 8 here, are differentially expressed in all patients, but at most L, right? So let's say we put L to be 20% of the number of patients. So this would translate also into 8 here, but that is coincidence, okay? So in other words, if you run Key Pathway Miner on such a data set, right, it will try to identify a subnet, a maximal connected subnetwork in here, where most of the Huntington's disease patients, where every gene but k exceptions are differentially expressed in all but at most 20% of the Huntington's disease patients, right? So if you set k to 8 here, then we have 8 so-called exception genes. So that is the red guys here, right? So this is the genes that are not differentially expressed significantly in most Huntington's disease patients, right? And if you know a little bit about Huntington, then you know that Huntington is a Mendelian disease. It's caused by one gene. It's a monogenetic disease, yeah, and it's caused by the Huntington gene, short HTT, and you can see here that it's right in the middle, right? So just for completeness, I also like highlight here the other genes that are known to be Huntington's disease related, but most striking is that we have this HTT gene here. So interestingly, this HTT gene itself is not significantly differentially expressed in most of the Huntington's disease patients compared to the control group, right? However, it is surrounded by many genes that are, because they react in their expression to a genetic variant in this gene, right? So even if you're using inappropriate technology, we're using gene expression data to narrow down on this disease, on Huntington, right? You can pull the causative gene out of the noise, right? In this case, the Huntington gene, right? Yeah, and also identify other genes. So this is kind of like the idea behind disease mechanism extraction. So we wrote papers about this, and it's used in studies and so on, and you can just find it by just Google for Key Pathway Miner. It will be the first hit. Now, with Key Pathway Miner, you still have the problem that the first thing you do is you analyze the statistics data, right? So like you first check if genes are significantly differentially expressed in most of the Huntington's disease patients compared to the control group. So in other words, we still work on genes that need to be significant individually, right? So KeePathway works on genes that need to be significant individually. And afterwards, we put essentially this signal on a graph, right? And then we check if the significant genes are randomly distributed over the network, or the protein-protein interaction network, or gene regulatory network, right? So we ask if significant genes are randomly distributed or if they enrich somewhere, right? However, this still has the drawback that each of the genes needs to be significant by itself. So we wanted to move away from this individual significance and move towards ensemble significance, right? So here we have a new tool developed, right? It's called Grand Forest and essentially means graph-based random forest. So random forest is a classifier. I hope that many of you have heard about this. So essentially, a random forest learns many such decision trees, then called a forest. Okay, so you have variables here, and you ask whether this variable is higher or smaller than a certain value, right? If it's higher, then you go right; lower, you go left. That is a typical decision tree. Now, in standard random forest, you can pick any kinds of features to go with these decision trees, right? So the only thing we actually do with our graph-based random forest, we restrict the way these features here are selected. So when you start with a gene 1, vertex 1 here, like this one, you can now for the next level not pick any other feature, right? That gives you the best split or best Gini impurity or whatever, right? No. You can only pick, or you can only select amongst those features here for the next level that are adjacent to previously picked genes. So in other words, when you're now with V1, here in the next level, here you can only pick amongst V3, V5, and V2. So this way, if you restrict it this way, every of the decision trees inside such a random forest automatically spans a subnetwork in a given graph, right? And of course, this such a graph here is again a PPI network, protein-protein interaction network, a gene regulatory network, any signaling network, any other kind of biological network you want to work with. Okay, so what we are doing here with Grand Forest is we are learning decision trees that automatically, by design, induce a connected subnetwork in a given input network. Right? And yeah, then you can do we have many such decision trees, right? So you can then run your standard analysis the standard analysis that you run with random forest, right? So like a feature extraction by mean reduction of gene index, for example. Then you get the most important genes, and you can check how they are connected. So this is something that you see here on the left side, right? So this is a typical supervised analysis, right? So that is something that you can do with Grand Forest. So you find the URL here, and it's published in Network and Systems Medicine. What it can also do, but I lack the time to explain how this works, is an unsupervised analysis. So we only show you like now how the supervised analysis works, right? So you can also apply this in an unsupervised fashion where you don't have like case control, right? So you just have a data set for a group of patients, for example, right? And you assume subtypes in there, right? And you want to explain each of these splits here with the mechanism, with such a subnetwork, and at the same time you want the subnetwork to predict good from bad outcome. So this is a bit more complicated, and we also have different tools. So unfortunately I couldn't be with you in Kharkiv today, but I will be in Kyiv next week. The whole week I will arrive on Saturday and stay with you the whole week. So if you're interested in how this unsupervised stuff works, just ask me. So we have a couple of tools developed here to address this problem, because it's significantly more interesting, but also significantly harder to explain in a short talk. So that's why we concentrate on this supervised part here. So what we did is we used our Grand Forest tool, and how do you show that something works in practice, right? So, well, like the intuitive way is, well, you compare it to knowledge that you already have, right? And want to show that you are able to like re-predict what you already know, and you only predict little extra, because then the little extra is more likely to be true as well. So what we did is here we downloaded lists of disease-associated pathways. There's different databases for this, right? For a breast cancer gene expression data set, lung cancer, ulcerative colitis, Huntington's disease, and ALS. So for like these 5 diseases, we downloaded gene sets essentially, right? So we know that they are problematic, but hey, so we downloaded gene sets where in the literature there's like a big body of research that essentially went into something like MCTB, but there's other databases for this that work a bit better, where where we have known disease-associated pathways. So, right, and we want to check if Grand Forest can predict many of the known disease-related pathways, right? And of course, we wanted to compare our Grand Forest GF here against other tools, right? Including our key pathway miner tool. That was the first one that I've shown you, right? And we wanted to compare it against our own tool. So the first thing you see is that Grand Forest outperforms all other tools, right? So including our own key pathway miner method, right? So our key pathway miner method works as well as Giga, Bionet, and GXNA, right? However, our Grand Forest approach outperforms the other typical de novo network enrichment tools significantly, at least with respect to the enrichment of known disease-associated pathways, right? So next thing we checked is for the 2 cancer data sets here, breast cancer and lung cancer. This is breast cancer, this is lung cancer. We checked if Grand Forest identifies more of the known cancer hallmark genes, right? So, and yeah, we do. So Grand Forest also outperforms all the other tools with respect to identifying cancer hallmark genes, at least with most of the cancer hallmarks, not with all of them, but with most of them. Okay. So what we then asked our question is like, so how come, I mean, we were happy, right? I mean, we assumed the ensemble approach to work better, but like we didn't assume it to work that much better, right? So like we didn't really trust our own results, right? So we were like, okay, so something, I mean, we already did all the randomizations here. You can see the Bok plots and so on. We did permutation tests, we did bootstrapping and so on. So like, I didn't trust the results. So what I asked Simon to do was then to do 2 checks. Like the first check was to check if the results that we deliver, so if we play with the size of the pathways that we want to report from like what small, like 20 genes to bigger pathways, right? That is the size of the networks, the subnetworks that we report here, right? So we vary this. We can vary this with this K parameter, then they're getting bigger, right? And you can do this with the other tools as well, right? So you have a parameter that you can play with such that they can give you bigger subnetworks as results, right? And well, I asked Simon to play with this parameter and to check how many of the genes in our predictions are statistically significant by themselves, right? So like how many of the genes, what is the percentage of the genes that are already by themselves predictive for the phenotype, right? Or in this case, predictive for the outcome, right? So then you see that with all tools, including our key pathway miner, right? Pretty much all of the genes in the solution, right, are statistically significantly predicting the outcome, right? I mean, this is also clear because that is how we selected them, right? So like we first pick genes that are significant by themselves, and then we check if they are enriched in a pathway somewhere. While Grand Forest, in Grand Forest, this is not the case. We don't work this way. So like we identify genes that only as an ensemble together become predictive for phenotype, right? And that's why we have a way smaller number of genes in our solutions that are predictive by themselves, but together they are more predictive than the individual ones that are predictive individually taken together, right? So that is what you see here. So, and then just for a control as a sanity checker, I asked Simon to do it the other way around, right? So I asked him, okay, please take the let's say a number of genes that we identify, right? So, or like, sorry, the other way around. So take every disease, let's say breast cancer, lung cancer, and so on, right? And check the disease pathways, take the disease pathways that we have used here, right? Take the genes in these disease pathways, right? So this is the known disease pathway genes, right? And check how many of those genes are, or check the fraction of genes that are already significant by themselves. And that is what you see here, for example. So the red guys are significant, the gray guys are not. In other words, on the pathway level, but also on the gene level, right? Most of the known disease genes are not statistically significantly associated with the outcome themselves. I mean, there's still a high number, sometimes a very high number, right? And I mean, here, if you look at this, right, or like these numbers, then of course, like if you pick well, even here, breast cancer, if you have 3,500 genes that are significantly different and good versus bad outcome, then of course, if you pick a random number of 50 genes, you have a very high likelihood of picking one or two or even more of those, right? That significantly associate with the outcome, right? But that is, but that's not really what we want to know, right? So we want to get down to the disease mechanism, right? So long story short, what have we learned? First, we have learned that disease genes are often not differentially expressed. We have learned that differentially expressed genes are often not the disease genes, right? That has to do with the passengers, right? And we want to move from disease genes to disease pathways, or as we call it, mechanotypes, or de novo endophenotyping is this field sometimes called. So I essentially now call it mechanotyping, right? So because you want to work with disease mechanisms, that is like composite features, right, that are emerging from graphs, right? So just to round this up a little bit, well, like 1.5 years ago, when this little virus hit our planet, and yeah, we were all sent to home office. I essentially allowed, yeah, pretty much everyone in the lab to take 2 months off with me, and to like stop work on whatever project they're working on, and to work on something, yeah, to help me develop something that people can use to battle SARS-CoV-2 or COVID-19, essentially nowadays called, right? Everyone, we just call it Corona, right? So, and pretty much half of the lab essentially decided to join. Some people were like too close to finish their PhDs or whatever, right? But a bit more than half of the people in the lab joined me in this endeavor. So our approach, like that was something we started last year in March when the shit hit the fan. Please excuse my French. And so our point of action, or like our attack point was the host pathways, right? So like while most antiviral drugs target the viral proteins, or they target the host interaction proteins, that is the proteins that the virus needs to in order to get into the cell or to get out of the cell or whatever, right? So we thought about, okay, let's not attack the viral proteins or the interactors, right? But the host interactors, they also interact with other proteins on the host level, right? And we asked the question whether we cannot interfere with the host proteins, right? And then we wanted to do this for 2 reasons. Number one is for this site here, we didn't have to develop a new drug. There was just, we can work with known drugs. That is a field called as drug repurposing, number one. And number two, well, for the virus, it's much harder to evolutionarily develop resistance, because you don't target the host protein, you don't target the viral proteins or the viral interactors, but you target something deeper inside the host. And this has a chance to work because it's illusory to believe that a virus with just a handful of genes can actually regulate everything itself. So the virus is essentially hijacking host mechanisms, right? And the idea is to find a way to interact with these host mechanisms that the virus is trying to control in order to get better, whatever, reaction chambers to replicate and so on. Okay. So what we did back then is we started with data from Gordon et al. Back then it was just a preprint. It was later published in Nature. It's essentially APMS data, affinity purification with mass spec. And he identified a bit more than 300 virus host protein interactions. In addition, we used the IID network, which is the human PPI network, protein-protein interaction network, and we integrated about 52,000 drug protein pairs, right? So that is like for the 7,000 drugs where they target in the human proteome. And then back then we had 67 drugs in clinical trials already against COVID-19. So now it's way more, but back then that was what we had available back then. And we used GTEx data because we wanted tissue-specific gene expression level because we knew that the virus was attacking the epithelial lung cells first, right? So that's the first line of defense. So we wanted to specifically look at the genes that are expressed in epithelial lung cells, for example, right? So that was important. So at the end of the day, such an integrated network here emerges, right? So you have the viral proteins, you have the human protein-protein interactions, and you have the drugs that target here. So that's essentially what we have gathered from this data integration exercise, right? So now you have to select seeds, right? So you can select either viral seed proteins that could, for example, be the virus spike protein. You can select host seeds that could be the ACE protein, right? That, for example, that the virus binds to, or you can select drugs, right? Like chloroquine, hydroxychloroquine, remdesivir was like one of the hot targets back then. Now we have way more, right? So, okay. Or you can, of course, pick a mix of those, right? So at the end of the day, what you do by selecting seeds is, if you look at such an integrated network here, so you have a set of, in this case, let's say seed proteins selected, okay? And what you now try to do is you try to connect them somehow to identify a candidate mechanism. There's different ways. One could be key pathway miner, of course. Another way, a more traditional way would be you just do a Steiner tree approach, for example, right? So you essentially try to find any kind of minimal spanning tree or something that is connecting all those guys, right? Then, of course, you might have several solutions, right? So of the same size. That is something that you often see with Steiner tree approaches when you have a network of a small diameter, like we have here in the PPI network. And then you combine them. So that's why we just call this here multi-Steiner tree or MUST, right? Well, but at the end of the day, what you receive is you have like your seeds, that is the dark blue guys, right? They are connected by light blue guys, and somehow they are somehow related with this viral infection. And then, of course, the intuitive idea is that these guys here in the middle, right? These connectors are, at least if you know drugs that target these guys, that are potential candidates for drug screening, or not drug screening, but for like in vitro drug testing and so on, right? So that's kind of like our drug candidates that we predict. So we have developed a tool that's called COVAX, like Coronavirus Explorer, right? That essentially does this. So I have just shown you this multi-Steiner tree approach, but we also have other tools here. Whatever between the centrality, key pathway miner, I have also shown you before, right? But at the end of the day, what you need is any kind of nice tool that allows you to select such seeds, right? Click a couple of buttons to extract such candidate mechanisms, and then click another button to extract approved or non-approved drugs, to show them in the network, compare this between SARS-CoV-1, SARS-CoV-2, MERS, and so on, right? So that is essentially what COVAX does. So if you're interested in how COVAX works, check it out. It's available here at this URL. And it's essentially implementing such a typical network approach. So that is how it looks. It comes as a as a web interface, and I mean, we would have never done it like this. Usually we would have implemented this as a Cytoscape plugin or something like that, or as a drugstore application. But yeah, back then we wanted something that every MD on the planet could just use with a couple of mouse clicks, something very intuitive with like a big quick start button and so on, right? And that's why we developed it as a web interface, right? So just a little example, right? So that was one of the case stories that we came up with back then, just to show you that what we predict essentially makes sense in a way, right? So let's say you have as observation as starting point to like select your seeds. You know that COVID patients have thromboembolic complications, right? So the hypothesis is that the virus interferes with coagulation pathways. So what you do is you select seed proteins. So here we select host proteins that are involved in coagulation. So we can use the integrated gene ontology feature by just looking for coagulation, and it selects those green guys here, right? Okay. So then on top of this, we select the viral proteins that interact with these 10 host proteins. That is these guys here. Okay, so that's my seeds. So next step is we look for drug targets by using this multi-steiner tree approach. So we try to connect these guys using the human PPI network. That is what we see. And if you have a closer look down here, you see that the coagulation pathway actually appears, right? If you now take this pathway here, these genes, and look for known drugs that target these genes here, you see these green drugs here. And you have hot candidates in here. That is these two guys, this PLAT and GGCX, that is these two genes. Why? Because they are known to regulate coagulation pathway genes, right? And they both interact with the viral proteins M and ORC2, right? So we have a potential mechanism linking infection with coagulation. So next thing you do is you do drug prioritization, right? So you look at these drugs here, right? And then you see these candidates here, these 5 hot candidates, because they are known for their anticoagulant action, and they are some of them interact. All of them interact with our hot target proteins here, right? So like these drugs, they target PLAT and GGCX, and they are known for their anticoagulant action. Okay, so this was just an like a little a little story about how you can use all this stuff and how you can do this quickly. So this is like part of the COVAX team back then. So we didn't all fit on like one slide, but yeah, it took us about 4 weeks to develop this, and then maybe another two months to get everything published and to go through the user stories and so on. So just to give you an idea about like how long things can take if you really want. So yeah, so back then it was a hot time. So nowadays you wouldn't make it into a hot journal with such an analysis anymore. But back then, I mean, we have been the very first ones to play with the systems medicine in SARS-CoV, and yeah. Our little hackathon ended up in a nice publication, which I show you not to show that we have publications, but to show you that the stuff we develop every now and then is actually useful and used for something, and that we can also have fun even when we are all locked into home office. Okay, so I showed you 3 tools essentially. One is Key Pathway Miner, and that, well, it works with omics. It extracts from omics data pathways. I have only shown you how it works with gene expression data, but you have to believe me, it also works multi-omics. Then I've shown you Grand Forest. That only works single omics, but it extracts mechanotypes. Is there any alarm? I think it's not me, right? Is there someone receiving a call? I think there was. Do you still hear me?
Yeah.
Yes.
Okay, good. Okay, so Grand Forest for mechanotyping, and then this COVAX system, the Coronavirus Explorer, essentially closes the gap between mechanotyping and drug repurposing. So that is essentially why I wanted to have it done back then, right? So I mean, it it was good that we could provide the community something to work with in the fight against Corona. But at the end of the day, I just wanted to see if there is a well-working systematic way of closing the gap between mechanotyping and drug repurposing. And I mean, the answer is yes, there is. So we have now a couple of PhD students working on top of this observation that it's actually doable. So that was pretty much what we did back then. So, of course, nothing works without funding. So, but more importantly, nothing works without people, right? So that is the ones that work the most on the stuff that I've shown you. Markus is now an associate professor at TU Munich, and Julian Maczynski is a PhD student in my lab, as well as Sepi. They are both about to finish. So Markus was the co-developer of Key Pathway Miner. Julian was the co-developer of COVAX. Sepi is involved in everything that has anything to do with systems medicine in my lab nowadays. She is working on something that is soon to appear. I didn't show it explicitly, but she's working on all this multi-Steiner tree things and so on. She was also involved in COVAX in Grand Forest. And Simon is now postdoc in Copenhagen. He is the co-developer of Grand Forest and the mastermind behind our Arnold Schwarzenegger-based prediction of breast cancer survival. That is the labs, right? So that was like a picture from the retreat we had, from the last retreat we had before COVAX, the last, a postdoc retreat at least. And that is like mainly the postdocs and senior PhD students. I see some, yeah. So that we had here in a little castle in the Alps. So here's more like the whole lab. So if you feel like squeezing into like a big group that barely fits into their own seminar room, then send me an email, but I'm happy to chat with you. So we are available at cozy.bio. And I'm grateful that you listened, and I have a second talk prepared. And I now like to know whether you want to ask questions now or whether I should first talk about the privacy aspects of all this, which is a shorter talk.
So please ask him.
So please, if you have and if you have any questions, you just approach the microphone and ask. Because we have only one. Please.
How about Pikachu gene?
Come again?
Pikachu gene.
Ah, Pikachu. Yeah, where's Pikachu star 5? Where do I have it? Pikachu star 5. And I have it here. So Pikachu star 5 is actually easy. In Pikachu star 5, you take the word Pikachu. So he called this an in silico mouse model. So in Pikachu star 5, he took the word Pikachu and just used the ASCII numbers of P, of I, and so on, right? And multiplied them by 5. And then that was the genes he extracted. And I can assure you, that was also statistically significant. And the other one was HP40. So what he did there is he took he went to Google and entered number of words in Harry Potter books. So that gives you, I don't know, I think 8 Harry Potter books or 7 Harry Potter books, right? And it gives you the number of number of words in each of these Harry Potter books. And he divided them by 40 modulo the number of genes in the genome, right? So that, in other words, gives you for 7 random numbers between 1 and 25,000. So and again that was significant. Yeah. I promised to Kyiv I will bring these slides. So I don't have them in the supplement here. I will move them, but I can show you next time.
Thanks.
Next week.
I think we will be able to share it with you also, because you are not participating in Kyiv, but only in Kharkiv. Yeah. Please, more questions, and please come closer to the microphone in order to say loud.
So if there's no more question, I would just continue.
Dear Professor, thank you for the interesting presentation. Could you tell me, please, if I understand you use random forest for feature selection? If I am correct, could you please tell me why? Why these methods?
Here, that part you mean, right?
Mm-hmm.
Yeah, I don't use it for, yeah, in a way I use it for feature selection. I use it for prediction. So what I want is I don't want to predict any outcome with just like a random set of genes, right? Or like a random set of genes. I don't want to predict with a set of genes, but I want to predict with a subnetwork, right? So I needed a method. Can I annotate here? Yeah, I can. Right? So I wanted a method. Whoops. And here we go. Better, right? So I wanted a method that extracts such subnetworks, right? So I wanted a method that by design can only pick subnetworks. So like the easiest way back then, I mean, you could probably also use geometric deep learning nowadays if you had enough data available, right? So if you do it with single cell data, and I can already tell you, like if you work with single cell RNA-seq data, we indeed work with geometric deep learning, which is a generalization of graph-based deep learning. But then you need very, very rich data sets, right? So like in single cell RNA-seq data, depending on what you look at, if you have single cell RNA-seq for like many patients, like 50 plus, you can actually use geometric deep learning, then you increase the precision. But back then, we have not been there, so we had to use bulk RNA-seq data. And that's why we used random forest, because it gives you like, it works on these decision trees here, right? So that was the only reason why we did random forest here, right? So we needed a method that we can modify to, by design, extract subnetworks. Yeah.
Thank you very much.
Sure.
Yeah, it's here.
And also, I would like to ask you about this program, Green Forest. Is it used only for human, or there are other organisms for which this database could be used?
Yeah, so Grand Forest, the online version works on, I think, human. I would need to check. I think the online version only works with human. I would need to check myself, I must confess. So the online version works only with human. However, you can download it, install it yourself, and you can plug in. Oh, no, sorry. Even for the online version, you can use any network. So long story short, the way it is, it only works with human. However, if you want to use whatever mouse, Arabidopsis, any other network, you can use it, but then you have to upload the PPI network yourself, which means essentially you need to go to BioGrid, download the network as GraphML file, and then you can upload it into, you can tell Grand Forest not to use the human PPI network, but to use a user-supplied network, and then you can upload the mouse, whatever gene regulatory network or PPI network or Arabidopsis or whatever you're working on, you can provide it yourself. So the interface allows this. Yeah, so that should work.
Okay, thank you for your answer. And one more question. And data that we gain after this Green Forest program, are they statistically significant? Or
Yeah,
do we
so
have a point of verification more?
yes, yes. So like the stuff that you see here, like this kind of things can be done automatically. So Grand Forest can give you this, right? And what Grand Forest also gives you, I don't have slides about this, do I? I think I don't have slides to show this, but no, I don't have
But you can tell us.
Yeah. Okay. Yes. So what it does is what Grand Forest does is if you have like a sheet where you can like click on, and then it does the verification, right? So model verification, right? essentially what you do is you randomize the class labels. So you do a permutation test. You randomize the class labels, and then you run the exact same pipeline again. And you can do this whatever 100 times, right? And then you can just count how often for each of these genes, right? You can just count how often do you see the genes in a randomly or in a random selection of the class labels, right? So essentially, you do a classical permutation test and just check if or you do a random a classical robustness analysis, essentially, right? So you check how robust your data is, right? So like in this way, you can give your solution a p-value if you want, right? So you can give even each individual gene in the mechanism a p-value if you want. And then you can colorize them by p-value, and then pick only those that don't show up in random solutions too often. So in this way, yes, it gives you empirical p-values. Yeah, but like the random forest itself doesn't give you p-values.
Okay, thank you very much.
Thank you. So more questions from audience? Doesn't seem to be the case. So
then I will continue quickly. Second part of the talk is shorter.
Yeah, I can understand.
Shall I?
Yeah, please.
Okay, cool, good. So then I've already introduced myself. So the big problem that we have with everything that I told you is that we need data, right? And this data usually, in particular when it's clinical data, needs to end up in any kind of cloud, right? So like you all have heard of the Amazon cloud or Google cloud or whatever. So we have tons of clouds. So there's an EU cloud, there's a Germany cloud. Every state in Germany has their own cloud. Every university has an own cloud. So big problem here is that, well, when it's clinical data, at least in Europe, we have something that's called a GDPR. But even if it's not by law, I mean, it's an ethical question, right? So who can access the data, right? So like, would you donate your genome data and then don't know what happens with it? So probably not, at least I wouldn't, right? So God knows what people would do with it in 20 years, right? And yeah, the world can change quickly, right? So for example, I am born in Eastern Germany, and when the country, I was 10 when the wall came down, 10 years old. So already me as a comparably young guy of 40 years already knows two different political systems. When I talked to my grandma, she told me that she lived under 5 currencies with 3 dictatorships and so on, right? So you never know what happens, and well, like nowadays you want to make sure that your data ends at least not ends up in the wrong hands, right? So in the European Union, this is in a way a bit regulated, right? So there we have something called GDPR, and like it comes with a couple of regulations, but the most important one is that patients must be able to revoke previously given consent, which essentially means the data must not leave the hospitals, right? So in particular, if your data, in particular genetics data and so on, if it ends up on any cloud, right? In particular, when the cloud is running outside the European Union, right? The doctor who is responsible to make things happen like this, right? If to revoke a previously given consent effectively happen, has no legal means of making sure that his patient's data or her patient's data is removed from the Apple cloud running in wherever, right? Or the Google cloud or Amazon cloud or whatever cloud you want, right? So this is getting problematic. So at the end of the day, this makes in particular MDs, like medical doctors, but also patients very, very, very reluctant, right, in allowing their data to be used in artificial intelligence in medicine, right? So, and that is problematic. So I'm coordinator of a big project, a big European project called Feature Cloud. So, and instead of working on like policies and traditional privacy enhancing technologies like better encryption, we work on something that's nowadays dubbed into privacy by design, or privacy by architecture, or some people now call it like dignity by design, essentially, right? So what you try is you try to like involve, think about privacy and data protection right from the start, right? So while at the moment data, in order to learn an AI, data needs to be copied into one cloud here, right? The idea is that you keep the data locally. You don't copy it to a cloud like it is here, right? But you keep the data locally. You learn a local predictive model, right? And then you just combine the model parameters into one big model. So essentially, instead of copying the raw data like genetics data, transcriptomics data, and so on to a cloud, right? You only communicate model parameters to the internet, which are much, much, much less privacy sensitive. So I'm not saying that they are always safe, sure. So there's like sometimes more, sometimes less theoretical side residual risks, right? So, but there's no 100% safety in life. But nevertheless, this field is called federated learning because the data remains, or like you learn the data remains distributed and you learn in a federated fashion. You can imagine this as an AI traveling from hospital to hospital and grow this way, or by you learn small AIs and combine them into one big AI, right? So this field is called federated learning. Now you might say, okay, hey, come on, okay, for genetics data, I see the point, or when I use imaging, right, in particular of the face or the skull or something, right? Like X-ray or skull X-ray is, of course, identifying. But hey, this is essentially not the case for like proteomics or metabolomics data. Yeah, I agree. However, usually what you need is you need meta information, right? So at least age, gender is information that you always need to correct for, right? So like typical confounding factors. Batch effects need to be corrected for. And very often you need to correct for population structures. In particular, if you want to come up with models that learn from data gathered in Europe, in Asia, in Africa, in America, and so on, the population structure is different, right? And you need to account for this, right? And the latest then, the data is becoming identifying, right? So because you need all this meta information, right? So it's very obvious that this data is becoming identifying when you also need comorbidity profiles and so on, right? So and electronic health record data and such, right? So then it's getting tricky. Okay, so in federated learning, essentially, you try to have any kind of decentralized way, right? And the way you can imagine this is with such a simple illustration here of for a linear regression. I know it's not a perfect predictive model, right? But it's I needed something easy that explains the idea, right? So let's say you have two parameters that you measure in 4 hospitals, one on the X, one on the Y axis, could be whatever size and weight, right? And what you can do is, if you want to learn a linear regression model, you can copy all the data into the cloud, right? And then whatever I use least squared method to compute like the slope of this line here, which is m, and then a parameter b, which is where this line crosses the x or the y axis, right? So we need these two parameters, right? Axis crossing and the slope, right? Okay. Well, like an easy way to do this in a federated fashion as well, like you just compute this locally, these m and b values, just copy these two parameters, m and b, into the cloud, right? And then compute the weighted average of these two parameters, right? And then you will have the exact same results, right? But you didn't have to compute any raw data. You didn't have to communicate any raw data from the hospitals to the cloud, right? And here it's quite obvious that this works, right? So now let's get a little more complicated. And I picked one example, and this is genome-wide association studies in GWAS, right? So in GWAS, what you do is you have a typical case control study, usually, not always, but usually, right? So you have a set of cases, you have a set of controls, right? And you try to identify in your genome loci where you have a so-called SNP, a single nucleotide polymorphism, where essentially like one position is like different in this group compared to the other group, right? And you this is called genome-wide associations because you associate a location in the genome with any kind of phenotype, right? A disease, for example. Okay, so that's called GWAS analysis. Okay, and then there's a set of typical statistics that you can apply, like a chi-square test or a linear regression or a logistic regression, for example. They all give you a p-value at the end of the day for every location, and then you need to correct this for multiple testing. You use whatever Bonferroni or Benjamini and Hochberg. Here, usually use Bonferroni correction. And then you can take the minus log 10 of this p-value and then plot these p-values here that give you such a Manhattan plot here, right? And then you can make such a line for genome-wide significance. And whenever you're above this line, that means your p-value is below whatever 0.05 divided by the number of loci, right? For Bonferroni correction. Then you have a significant location, whatever here in chromosome 6 at this location or in chromosome 8 at this location, that is in a way predictive for the phenotype, or as we say, associated with the phenotype. And you usually have to account again for confounding factors like age, gender, and so on, but you also have to correct for population structure. So the standard analysis tool is called P-Link here, right? So like it's working centralized, so it's very robust to imbalanced data. So if you have hospitals where the data is very imbalanced, so that doesn't have to be like the case and controls that have to be imbalanced. It can easily be any kind of confounding factors that are imbalanced or a different population structure, right? So in either way, when you have this information and when you can use it with P-Link, so you can provide very robust results. However, P-Link requires the data to be like available on one central hard drive, essentially, which means it's not privacy preserving. And because this is problematic, and there's tons of GWAS studies where the data is gathered across different countries and so on. Well, like in these countries, they run essentially their P-Link separately, and then they do something that's called meta-analysis. So you just take the p-values and the scores for every SNP, right, or for every loci, and then you combine this in a way. So there's different ways of doing this. There's different tools, right, for meta-analysis, and they are privacy preserving. However, they are not very robust to imbalance data, as I will show you, right? So if you have heterogeneously distributed confounding factors or like cases, for example, right, then these tools are problematic. And we have developed something that's called splink, like secure P-Link, which is giving you the exact same results, but without having the privacy problems, right? So that's our contribution here. So just for an illustration, right? So here it's for COPD, risk factor for lung cancer, right? Chronic obstructive pulmonary disease. We have 2,800 COPD patients, 2,500 in a control group. We only checked for 680,000 SNPs, and we only use sex as confounding factor. So very easy. So like just to stay easy now for the presentation, okay? And the first thing we did is we split the data set here into 10 subsets to simulate that it was gathered at 10 hospitals. And we only use logistic regression. Again, that's the only result I will show you here just for illustration, right? And then we can compare the centralized p-values and beta values, which is the score, and the p-value for that score, right, with the federated values and the meta-analysis. So first thing is centralized versus federated, and I can show you here in a bit also with the Manhattan plot that it is the exact same results for the centralized analysis and the federated analysis, right? So here the exact same p-values, we have the exact same results. However, in meta-analysis, you can see in particular in the areas around the threshold, this is getting problematic. I know it's not a very systematic study here. This is just for illustration, right? How you can test such models, right? So you see that the P-Link based meta-analysis with a fixed effect model just doesn't perform so well. So the idea is that we now checked, compared the balanced versus the imbalanced scenario here, right? And in the balanced scenario, you have like the confounding factors and the case control groups and so on, like distributed in the same way. So this is what we did now. This is what I did here with COPD, right? So we randomly split into 10 subsets, right? So just uniformly distributed. But you can also do this in an imbalance. And then, of course, what you see is that you have a high correlation between the P-Link p-value, right, which is the gold standard, and our splink p-value, which is the blue guys here, and our meta-analysis p, which is the red guys, right? So, but you can provide an imbalance scenario, right? So where you like heterogeneously distribute factors, right? And then when you see this comparison, you will see that our S-Blink and P-Link still give similar results, while the meta-analysis tools just dropped drastically in classification performance. So first thing we distributed heterogeneously was the phenotype, then gender, then smoking status, right? And then also age and smoking pack years, we can also distribute heterogeneously, right? So then we come up with different classes, right? So we call the data either balanced or slightly imbalanced or highly imbalanced or whatever. And also we can have a heterogeneously hidden confounding factor, right? Which is the yellow one here, right? And then we can compare our S-Blink, our tools, like the different meta-analysis tools, that is those here, and our S-Blink tool against the result we would have with the centralized analysis with P-Link, right? And then we can check the correlation of p-values of our S-Blink tool with the p-values of our P-Link tool. And then you see that you have a correlation of 1. However, with the and that is independent of how imbalanced and how heterogeneously distributed the data is. While with all meta-analysis tools, you can see that the classification performance drops. If you now not just check the correlation, but if you really check for the COPD gene data set at least, the number of correctly identified SNPs, right? Then you see that for severely imbalanced data and for data with heterogeneously distributed confounding factors, you have no signal anymore at all. So you lose the complete signal, while our S-Blink tool is still identifying everything. And we have verified this these observations with an independent data set. So we have to just use FinGen and extracted COPD data from FinGen to just show that we see the same picture. That is not an observation bias from our COPD gene data set. So this S-Blink tool is available here at this URL, and it's running standalone. And when you go to this S-Blink, you have a little video here that explains the concept and explains how to use it and why it's privacy preserving. So, but that was just an example. So we have actually developed an AI store, really like a whole system working, providing different tools, templates, and so on to run such federated AIs as kind of apps in a federated fashion. It's called Feature Cloud or the Feature Cloud AI store, right? So you can find it at featurecloud.ai. It comes with we already have more than 25 or 30 such apps. So I've just shown you like this GWAS app here. And I think here like linear regression, the example, but we have tons of more like AdaBoost and a random forest regression. We have a deep learner and so on. And I mean, it is an app store. It is extendable. Everyone can go there. It is completely open. So you can develop federated machine learning apps and put them into the app store, and then people can run them. It comes with like different additional security measures like communication scheme based on secure multi-party computation. You can have like standard privacy enhancing technologies like differential privacy, right? And then from the application point of view, you have any kind of coordinator running a study. Coordinator creates a project, essentially plugs a workflow together so you can run several apps in one workflow. Then the coordinator can invite participants, they can join, they can also download then the learning app, right? They all feed it with their own data. Then the federated learning is going on, and then one big model is learned and provided to the coordinator afterwards, and then the coordinator can do with it whatever he or she wants. We don't need to, I will skip this. That one is just to show that we can have, we can plug such apps, for example, for like importing, batch effect correction, then you do your regression models or whatever you want to predict. Then any kind of output and visualization, right? You can put them in one workflow system, in one pipeline. I've just shown you like this GWAS thing here, right? We already have apps that is the most low-hanging fruits here for gene expression data analysis. The standard tool is called limma. So we have provided now a tool called Flimmer, like federated limma. Here's just links to the preprints. And there's a tool called Party, like just to show you, just come back to these survival analysis, these Kaplan-Meier plots here. That is also something that is now working in a fully federated privacy-preserving fashion. So here's just a list of such like a couple of first tools that are available. Some of them are also available as standalone tools. Some of them are only available as feature cloud apps. Yeah, that's the Feature Cloud Consortium. That is the PIs and the Feature Cloud Consortium. I just happen to be the coordinator. So there's many people I could thank here. If you find all of us, then go not to featurecloud.ai, but .eu. Right? Then you find the consortium. Again, our lab. And if you're interested in following up on this, both for like Flimmer and Esplink, we provide little videos explaining like the overall concept, but the easiest way of these two tools, and like showing click by click how you can reconstruct the results yourself that I have just shown you. But the easiest way is you guys just go to featurecloud.ai and then watch a little 3-minute video explaining the overall concept of federated learning, why it's privacy preserving if you do it the right way, and how to use the Feature Cloud App Store and how to get started. And we have a couple of more videos there explaining like really in 10 minutes how to like develop your first own federated AI apps and how to put them into the Feature Cloud App Store and so on. And that's about it. Thank you very much. And yeah, open for questions. I hope I didn't take too much time. Oh yeah, I overdid by 3 minutes. Sorry for this.
Thank you, Jan, for this interesting talk. We have now prepared outside a coffee session for our participants. And so I suggest that those people who have questions anyway should come close to the microphone, gather around the microphone, continue a little bit the discussion with you for the next 10 minutes until we start our next session. And the others rush out, grab a coffee and whatever is available. I have no idea. And Jan, thank you again for giving your lecture. See you next week in Kyiv and potentially see you before in Chernobyl. I just sent you all the information and you need to respond very soon.
I will. Thank you very much.
So, anybody who has a question, please come to the microphone closer and discuss with Jan.
One option you can send your question via Hua. Actually, Professor Baumbach, did you download the Hua app?
Yes, I'm in Huva. Let me start this again. I am in there here. And yeah, I just received too many messages there informing me about new attendances. Not to me. It's just like, you know, a speaker and VIP chat room. And I was tagged in a photo. I don't know how this works because I haven't been around, but hey, you know. So I have 64 community messages. Break the ice, share your moments. So I will try my best, but then now I see. So I will try my best to like, yeah, check what I'm reading there. But I think if you send a message directly to me, I can actually, I will not miss it.
Okay.
I can see you took a picture of my slide there. Ha! Whoa! Now I know why I'm Kostianten did this. So thank you very much. Now I know why I'm tagged in the picture. Thank you.
It looks like it works.
Yeah, it looks like it works. Cool.
So, we have one question from a participant, Konstantin. Please.
Jan.
Hi.
Hi. You mentioned about the remdesivir.
Yes.
Could you say something about investigation of this for the COVID treatment?
Yeah, with remdesivir, it was like this. We also rediscovered this, just like others did. So that is the short story, right? So number one thing we learned about remdesivir, if we pick drugs that are known to be effective, right? Or like in a way effective, right? So back then we didn't know much, right? So we just pick chloroquine, hydroxychloroquine, remdesivir, right? Check what they target, check if they, right? But they essentially target like directly the interactors, right? So so then you don't gain any additional knowledge, right? So, and that's also how they were picked, right? So if we do it the other way around, like with the coagulation story that I have just shown you somewhere here, right? If you start like this, you also read this, not with the coagulation story, but we have a couple of other strategies of identifying seed proteins not based on thromboembolic complications. Then you indeed see or rediscover remdesivir. So that was the only reason why I mentioned it. Was that you can either start with something that you know is effective and you want to identify more, or you want to understand where it's targeting, right, and why this could be effective. So this became in particular important when people had tons of high throughput screening studies running, right? So like back then, beginning, there was an infinite amount of money available for doing high throughput screens, right? So we had like infected cells, and they just wanted to see if there's any
I
drugs.
understand.
Yeah.
I understand. So thank you.
Okay.
Sure.
Thank you, John. We have now to continue with
Yes.
the next session, which is starting in 5 minutes and needs a bit more preparation because it's an online workshop. So thanks again and see you then next week.
Thank you.
Bye-bye.